Friends, welcome back to my channel, and today in this video we are going to study about conjugation. In this we'll study about other gene transfer mechanisms also. We'll learn about auxotrophs and prototropes, Lederberg and Tatum experiment, Bernard-Davis experiment, what is the difference between F plus, F minus, HFR and F prime cells, and we'll also study about sex reduction. So now we'll study about the methods of gene transfer. Now, there are two ways by which gene can be transferred, that is vertical way or vertical method, and another one is horizontal method. As we all know, a cell divides into daughter cells and the chromosome in the parent cell will get transferred to the daughter cells of the next generations. So this we call it as a vertical transfer method. What do we mean by horizontal transfer method? When the DNA get transferred between the cells of same generation, then we call it as a horizontal transfer method. So under horizontal transfer methods, we have three ways by which the gene transfer can take place. That is conjugation, transformation and transduction. So what is conjugation? As you can see in the diagram, the conjugation, that is one cell will act as a donor cell and another cell will act as a recipient cell. Which cell will act as a donor cell? The cell which will have the F plasmid. Here you can see this F plasmid is present, whereas in the recipient cell, no such plasmid is present. Clear? So definitely, if you have to give a pen to your friend, definitely you should have that pen with you. If you don't have pen, how you will donate to any other person, right? Similarly for the bacteria also. Until they have the F plasmid, they cannot donate it to anybody else. So here, the first bacterium contain F plasmid. So this bacterium will act as a donor bacterium and it can transfer this F plasmid to the recipient bacterium which lacks the F plasmid. Now you can see for the donor to transfer into the recipient, what happened? A physical contact between both the cells. After the establishment of this physical contact, the F plasmid getting transferred to the recipient, and now the recipient also became a new donor because now it has this F plasmid which it lacked in the beginning. Clear? So now the donor has donated the F plasmid to the cell that lacks F plasmid, that is a recipient cell. So this method is known as conjugation because it happens due to physical contact between the two cells with the help of pilus. Another method is transformation. What do we mean by transformation? This is one bacterium cell. What will happen? It will donate its chromosome to the recipient cell. This chromosome will enter and it will bind with the chromosome of the recipient cell. So this method is called transformation. Here in transformation, as you can see, in both this bacteria, there has been no physical connection between them. Without the physical connection when the transfer happens, we call it as a transformation. Why it happens? Maybe due to some reason, this first bacteria died or it got degraded, because of which its DNA is now released in the medium. And because a DNA is present in close vicinity of the second bacteria, the second bacteria took this DNA inside its cell. So both will combine and now the yellow segment is integrated inside the red segment. So that's how when the transformation, when the gene transfer take place without any physical contact, we call it as a transformation. Transduction means it is the method when gene is getting transferred from one cell to another through a bacteriophage. What is bacteriophage? Bacteriophage are the viruses that attack bacteria. So what you can see here, this is one bacterium A. In that, any bacteriophage attack, and how the bacteriophage attack? It insert their DNA inside the bacterium cell. So now the bacteriophage DNA is inside the bacterium cell. Now the phage DNA will replicate. Along with that, the chromosome DNA will also get replicate and it will break down. Now what will happen? It will pack the DNA again so that it can get released from this cell and infect any other cell. So before it get released into the environment, it will pack the DNA. So while packing, it will pack its own DNA. But sometimes it happen like it pack the bacterial DNA. Now it get released into the environment and ready to attack any fresh or new bacterium. Correct? So when this particular bacteriophage which had packed the bacterial DNA will infect any another bacteria. This is second bacteria. So when this bacteriophage will attack this bacteria, how it will attack? It will insert its DNA inside the bacteria. So now the DNA, the black one, which is getting inserted into the second bacteria, this black one is actually the DNA of this first bacteria. Now this black one will get inserted into this red chromosome. That means the black one is the DNA from bacteria 1. So the DNA from bacteria 1 getting transferred to DNA on the second bacteria. So this method of gene transfer, we call it as a transduction. So we have three methods. Conjugation, that means through physical contact. We already saw the contact happened due to pilus. Transformation without any physical contact. Transduction with the help of bacteriophage. Now, what do we mean by auxotropes and prototropes? Auxotropes means that lack some essential elements which are needed for the growth. Prototropes means they consist of all the essential elements that are needed for the growth. So we have two kinds of medium, minimal medium and complete medium. Minimal medium means it will consist of medium where only prototropes can grow. That means it will only support the growth of prototropes that can manufacture all the essential elements. That media is known as minimal media. Complete media means when we add extra elements in the medium, when we know that the particular organism or bacteria cannot grow on the medium, so we will add extra elements on the medium for the growth. That media is known as complete media. So auxotropes means the bacteria that lacks some essential elements that are needed for growth. Whereas prototropes means they can synthesize all the essential elements. So on the minimal media, prototropes can only grow, auxotropes cannot grow because they lack. Until you provide in the media, they cannot grow. So auxotropes can only grow on complete media because you will provide that extra element which auxotropes cannot synthesize. Whereas minimal media means the word minimal. You will provide only that much substance in the medium that can be used by prototropes to synthesize all the necessary elements by itself. Now, come to Lederberg and Tatum experiment. What they did is, they worked with two strains of auxotropes. In auxotrop 1 or Y10 strain, it lacked threonine, leucine and thymine, whereas biotin, phenylalanine and cysteine is present. Whereas in the second auxotrop, first three is present, the next three is absent. Now, when they are placed on the minimal media, what you will expect? This auxotrop will not grow because as we learned that auxotrop cannot synthesize all the essential elements needed for the growth. And in this auxotropes, it cannot synthesize threonine, leucine and thymine. So it cannot grow. Whereas auxotrop 2 will also not grow on the minimal media because auxotrop 2 cannot synthesize biotin, phenylalanine and cysteine. But it was observed that when both the bacterial strains are added in one plate, we found some colonies grow. Now, why some colonies grow? They should not grow because half of the auxotropes lack threonine, leucine, thymine, half of the auxotropes lack biotin, phenylalanine and cysteine. So they should not grow. But we found some colonies. So that means the gene has been transferred either from auxotrop 1 to 2 or from 2 to 1. Because of which some colonies are positive for all the 6 essential elements. And the colonies that are positive for all the 6 essential elements able to grow in this medium. So what we can conclude from this experiment? That yes, genetic exchange or recombination took place between two mutant strains. Now then came Bernard Davis and he performed YouTube experiment. What's the experiment about? In a YouTube, minimal medium has been added. And on one side, auxotrop 1 is added. On the another side, auxotrop 2 is added. In between, a filter is attached which can allow medium to flow but not bacteria. So now, after some time, it was observed that no growth in the minimal media, either from this end or from this end or from here or from here. From wherever the cells has been plated on the medium, they observed no growth in the medium. Why? In the last experiment, we saw that they can exchange genetic material and that's why we observed some colonies growth. But here we are saying no growth has been observed. So what we can conclude with this? We can conclude that genetic exchange requires direct contact between bacterial cells. Because this filter is attached which does not allow bacteria to flow, hence the bacterial connection has not taken place. And because of lack of this connection, they remained auxotroph because no genetic exchange has taken place and hence no growth was observed in minimal media. So it is concluded that genetic exchange takes place between the cells of same generation, but for that direct contact is needed. Now, what do we mean by F plus and F minus cells? So as you can see here, this is a F plus cell. F plus means the cell that contains F plasmid, that is the donor cell. So the cell is a donor cell because it contains F plasmid and hence we call such cell as F plus cell. F minus means definitely the cells that does not contain F plasmid are known as F minus cells and these are recipient. So always the donor can pass the F plasmid to the recipient cell through a direct contact or physical contact between both the cells via pilus. Now as you can see here, ORT means origin. From here only the chromosome will break and it will get transferred to the recipient cell. So when it breaks from ORT, as you can see this red one, this is the segment which enters into the recipient at the first go. That means if this is a donor cell and blue one is the ORT, the chromosome will break from here. And how it will transfer? This blue one will move first. Suppose some genes are present A, B, C and D. So in the recipient bacterial cell, this blue one will move first. So what will happen? This blue one will insert first and after that, this rest of the chromosome will enter. So always the transfer of genetic material from one cell to another, it has a direction. Always it will enter from this ORT site. So whatever the gene that follows the ORT, in the same order, the gene will get transferred into the recipient cell. So after transfer, what will happen? The F minus will also become F plus cell. Whenever the gene is getting transferred from F plus to F minus cell, after transfer, this F minus cell will also become F plus cell. So this is only written here. Now what do we mean by HFR cell? HFR cell means, as you can see here, F plasmid, when enter into the recipient cell, sometimes it happens that instead of being present separately inside the cell, it gets inserted into the chromosome. Recombination happens between chromosomal DNA and this plasmid DNA and it gets inserted into the chromosome. Such cell is known as HFR cell. F plus cell, the cell having chromosomal DNA and F plasmid. F minus cell, the cell will having only chromosomal DNA and no F plasmid. When F plus cell donate its F plasmid to F minus cell, the plasmid will enter inside F minus cell and will remain present as an extra chromosomal DNA. So now this F minus cell also became F plus cell. But sometimes it happens like when it donate this DNA to F minus cell, during that time what happens is this F plasmid get integrated inside the chromosome of the recipient cell. Now this cell we call it as a HFR cell. Very less chance that this phenomenon occurs. This HFR phenomenon to occur, there is a very less chance. It is a rare case when such things happen. So mostly what it happens is, you can see in the diagram, it degrades. So the F minus cell will become HFR. The chance of this phenomenon to happen is very rare. Now one more thing is F prime cells. What do we mean by F prime cell? So as you can see in the diagram, this is an HFR cell. Sometimes what happens that crossing over takes place within this HFR cell only and again the cell becomes F plus type. But this we don't call it as a F plus, instead we call it as a F prime. Why? Because as you can see, when it gets separated as an extra chromosomal part, this gray region of the chromosome is also present here. So this is called as F prime cells. F prime cells means these are also F plus cells, but here as an extra chromosomal part, it contains not only the F plasmid, but also some portion of bacterial chromosomal DNA. In that condition, the F plus cell, we call it as a F prime cell. Now, when such cells, that is F prime cell, when combined with any other cell, and transfer its plasmid to the recipient cell, that method is known as sexduction. This is the summarized table for what all we studied. F plus cell, that means along with chromosomal DNA, we have F plasmid. F minus cell, only chromosomal DNA. HFR cells, in this chromosomal DNA only, this plasmid section is present. F prime cell, along with plasmid section, chromosomal part is present. So whenever there is a conjugation between F plus and F minus, finally we'll get 2F plus. Why? Because F minus also becomes F plus. Whenever there is a conjugation between HFR and F minus, we'll get 1F plus and 1F minus, because already I told you this is a very rare condition when any F minus cell becomes HFR cell. So whenever a recombination happens between HFR and F minus cell, we'll have 1F plus and 1F minus. If the conjugation happens between F prime and F minus cell, we'll get 2 F prime cells, because F minus will become F prime. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.